Welcome back. In this section of the course, we've been discussing how animals can respond to their environment and how information can be transmitted through these large multicellular bodies that are found in animals. And in the last web lecture, we had set the stage for this information transfer. We talked about how the resting potential is generated. We saw that ATP is used to store the potential energy in this electrochemical gradient across the cell membrane of the neuron. We saw how that resting potential can be altered by signals coming in from other neurons in this graded potential. And we briefly mentioned how once this threshold potential, this voltage of around negative 55 millivolts, at that point something different is going to happen, and this is called an action potential. And so we've seen this picture. In this web lecture, we're going to find out more about how this occurs and also how it's propagated all the way along an axon to be able to send a signal very quickly, much more quickly than would be possible using something like chemical messages. Even being propelled by the circulatory system is not going to be nearly as fast as this electrical transfer of information. So remember, this electrical signal, this spike in depolarization, is going to move all the way down the axon, and it's going to do it without losing any of its strength along the way, no matter how far down the axon it goes. So we're going to talk about how that happens. So all of this has to do with changes in the membrane potential. And so we can actually record these changes in membrane potential using equipment called microelectrodes, actually measuring the voltage inside the cell relative to outside the cell, and we can actually measure and record what's going on during these action potentials in terms of the membrane potential. So changes in membrane potential occur because neurons contain gated ion channels that open or close in response to stimuli, and opening and closing these gated ion channels is going to allow ions to flow across the membrane along their electrochemical gradient. In particular, voltage-gated ion channels are going to be very important in this action potential process, and they're going to open or close in response to a change in voltage across the plasma membrane of the neuron. So they're going to open when a certain threshold membrane potential has been reached. And we saw in the last web lecture how this comes about. The neuron is going to be getting different kinds of signals from other neurons that are going to be summed together. So remember, any individual stimulation is not going to be enough on its own. Multiple stimulations have to occur either in close succession coming from the same presynaptic neuron or close together in space. And they're going to sum together. And then there's also going to be the effect of these inhibitory signals that are going to make it less likely that an action potential is going to happen. And so the sum of all of these inputs, if it brings the voltage down to that threshold potential, that's when we're going to see this dramatic change in membrane potential. So a threshold depolarization is going to lead to an action potential every time in this all or nothing manner. So let's quickly compare a graded potential, which is what we get when all of these inputs are coming in around the dendrites and cell body, to an action potential, which is going to be initiated at the axon hillock and propagated down along the entire length of the axon. So graded potential is going to be the cell's response to ligand-gated channels. So remember, those neurotransmitters are going to be the ligands that are going to bind to their receptors on the postsynaptic neuron and open those ligand-gated channels. Action potentials are going to be due to the opening of voltage-gated channels when that threshold depolarization is reached. Graded potentials can sum together, and also the magnitude of the change in membrane potential is going to depend on the strength of the stimulus. So it's possible to have a greater or lesser depolarization in response to these graded potentials. In contrast, the action potential is all or nothing. It's always going to have the same strength. It's always going to take about the same amount of time. It's going to be an event that happens, and either it happens or it doesn't, much like our skydiving squirrel. Either it's going to jump out of the airplane or it's not going to, but there's nothing in between. In a graded potential, the amplitude depends on the strength of the stimulus. So that means that the degree of depolarization or hyperpolarization is going to depend on the strength of that stimulus. In contrast, an action potential, because it's all or nothing, each one of those signals is going to be exactly equal, but the intensity of the stimulus is going to be communicated by how often those action potentials come through. So the rate of firing is going to send the information about how strong that stimulus is. So what is an action potential? So an action potential is a rapid temporary change in a membrane potential, and it has three phases. The first is a depolarization phase in which the membrane becomes less negative and moves toward a positive charge. Then we have a stage of rapid repolarization, which is going to change the membrane back to a negative charge. And in fact, it's going to overshoot a little bit, and hyperpolarization will occur when the membrane becomes more negative than it was during the resting potential. So for an action potential to begin, the membrane must depolarize from the resting potential of about negative 70 millivolts to a threshold potential of somewhere around negative 55 millivolts. And this is the threshold potential that's going to cause those voltage-gated ion channels to open. If that threshold potential is not reached, the action potential won't occur. If it is reached, it will always occur. So how does this depolarization occur? An action potential results from changes in membrane potential as ions move through voltage-gated channels along their electrochemical gradient. So let's start with resting potential and see what happens. So at resting potential, most of these voltage-gated sodium and potassium channels are closed. So we see the channel is blocked here from the inside on all three. We've got this resting potential of around negative 70 millivolts, which is the result of the equilibrium potential of all of the ions in the cell. Then we begin the depolarization stage. So when an action potential is generated, the very first thing that happens is when that threshold is reached, that's going to trigger the opening first of the voltage-gated sodium channels. So this is going to cause sodium to flow into the cell at a very high rate. Why? Because remember, they have both a concentration gradient. They're highly concentrated on the outside of the cell. So they're going to move along their concentration gradient into the cell. And there's also this voltage gradient. They're going to be attracted to the negative charge inside the cell. So this is going to be a very rapid influx of sodium through these voltage-gated channels. At this point, the voltage-gated potassium channels are still closed. So we're just going to have this huge influx of sodium ions. So this is the depolarization phase. So as these sodium ions flow in along both their concentration gradient and their voltage gradient, the membrane is not just going to depolarize. So remember, it's depolarized when it comes to zero, but it's actually going to continue to increase toward the equilibrium potential for sodium, because remember, potassium is not yet flowing through, and so the greatest influence on the membrane potential is going to be this equilibrium potential for sodium as it flows into the cell. So we're going to go all the way up to about plus 40 millivolts. So it almost reaches but doesn't quite reach the equilibrium potential for sodium, because the next thing that happens is that those voltage-gated potassium channels open at this point. So at this point, there's actually an error in this diagram. This is going to be positive inside, right? Because we've got a membrane potential of positive 40. So this is actually, at this point, going to be moving both along its concentration gradient. Remember, potassium is highly concentrated inside the cell, and also now along its voltage gradient, because now it's going to be relatively more negative outside. And this is going to cause the repolarization of the membrane. You're going to bring a bunch more positive charges back out of the cell and cause the cell to repolarize back down to negative. At this point also, there's an inactivation of these sodium channels. So as long as the membrane potential is above this threshold, the channels will remain open, but they become blocked by a portion of the protein that inactivates that channel so that there's no actual movement of sodium through. So now the membrane as a whole is going to be moving toward the equilibrium potential of potassium because potassium is flowing and sodium is blocked from flowing. Because of this blocking, this inactivation of the sodium channels, this is actually going to undershoot that resting potential. Why? So remember that once the membrane potential gets down below this threshold potential, the voltage-gated channels are going to close up again. Once these voltage-gated potassium ion channels close, then there's just going to be the influence of the potassium and sodium leak channels, which are going to bring that membrane potential back up to the resting potential, ready for another action potential. So to summarize, this rising phase is due to the opening of voltage-gated sodium channels that begins when the threshold membrane potential is reached. There's going to be a huge rapid influx of sodium along both the concentration gradient and the voltage gradient, so sodium ions are going to rush in during this rising phase, this depolarization phase. When it gets to about plus 40 millivolts, then those potassium ion channels are going to open. There's going to be a huge efflux of potassium ions out of the cell along both their concentration and voltage gradient that's going to repolarize the cell membrane. At this point, the sodium ion channels are inactivated, and so the membrane potential is going to move toward the equilibrium potential of potassium. It's going to undershoot that resting potential. When it gets below the threshold, all of those voltage-gated channels are going to close, and so it's going to return back to its resting potential due to the equilibrium potentials associated with just the leak channels. So let's take a look at how this signal moves along the axon. So in addition to being fast and having 3 distinct phases, the action potential is an all or none event. So there's no such thing as a partial action potential. It's going to have the same magnitude, and it's going to move all the way along an axon every single time. So all action potentials for a given neuron are identical in magnitude and duration. And action potentials are going to be propagated down the entire length of the axon, and these axons can be very, very long. For example, in our bodies, we have a single axon that goes from the base of our spinal cord all the way down in a single axon down to the muscles in our toes. So we've got axons the length of our leg, and these action potentials are going to move all the way along them every single time. Neurons have excitable membranes because neurons are capable of generating these action potentials that propagate rapidly along the length of their axons. And this action potential suggests a mechanism for electrical signaling. This whole action potential process allows for it to be regenerated continuously along the entire length of the axon, and we'll see how that works. So in the nervous system, information is coded in the form of action potentials that travel along axons, and the nature of the information is coded by the frequency of action potentials and the pathway that they take through the nervous system. So how is this action potential propagated? How does it move along the axon? So the action potential is propagated down the axon in 3 steps. First, the influx of sodium ions at the start of an action potential repels all of the cations, all of the positively charged ions that are already there inside the cell. So that's going to cause them to move away from that site toward a more negative area. So this results in the charge spreading away from the sodium channels. As positive charges are pushed farther from the initial sodium channels, they're going to depolarize adjacent portions of the membrane because they're making the interior of the membrane less negative as they move toward those adjacent portions of the membrane, depolarizing them and moving them toward that threshold potential. So then, once they reach the threshold potential in the next section of the axon, those voltage-gated sodium channels are going to open, and that's going to result, again, in a full action potential at that point in the axon. And this is just going to repeat all the way down the axon. That depolarization, the influx of sodium ions, is going to spread out to the next little section of the axon, cause an action potential there, and so on and so on and so on, all the way along the axon. And so remember, this action potential at every part of the axon is all or nothing, so it's going to propagate this without diminishing in any way the amplitude or the strength of this action potential. So think of the action potential as being regenerated at every step in its entirety, in full strength. Once sodium channels have opened and closed, they become desensitized to depolarization, so they're less likely to open again for a short period of time, and this is known as the refractory state. The action potential moves only in one direction down the axon from the cell body out to the axon terminal. It can't move in the opposite direction because that previous section of the axon, even though those positive charges are moving in that direction, it's been inactivated, so it's not going to respond again. Only the part of it that hasn't previously responded is going to respond to that depolarization. So again, action potentials propagate only in one direction because the upstream sodium channels in the direction of the cell body are in that refractory state. They can no longer be stimulated for a short period of time. They'll be ready when the next action potential comes through, but the action potential cannot move backwards toward the cell body. So let's take a look at how this works. So here we've got a little section of an axon, and we see this voltage-gated sodium channel has opened, creating a whole bunch of positive charge right here next to it. That positive charge is going to repel other positive ions. It's going to attract negative ions, and that's going to cause the inside of the membrane here next to it to be more positive. It's going to be depolarized. It's going to trigger those voltage-gated sodium channels in this next portion of the membrane and cause an action potential there. So here we see the depolarization of this next ion channel a little bit further downstream, and this is going to trigger that action potential, opening up this voltage-gated channel. This one becomes inactivated, so it's not going to be sensitive to this inflow of positive charges in this direction, but whichever voltage-gated channel is a little bit further down will, because it hasn't already had an action potential, and so it's going to move just in this one direction. So if we were to measure the voltage at different sites along an axon, we'd see the action potential happening earlier here at point A, a little bit later at point B, and then a little bit later at point C, as this wave of depolarization moves along the axon. Looking at this a little bit differently, this is the figure from your book. Here we've got the action potential at this point on the axon. This positive charge is spreading out to the neighboring portions of the cell membrane, depolarizing them. So now this section is depolarized. This is repolarizing. Remember, the sodium channels are blocked, so the positive charge coming from this direction is not going to have an effect, but the positive charge going in this direction is going to depolarize this next section of the membrane and cause an action potential there. So this is the way that action potential is propagated in a wave all the way down the length of the axon. And remember, this is an action potential that's being constantly regenerated, so it's not going to lose any strength as it moves along. It can go down the entire length of the axon, no matter how long it is. Now let's take a look at some of the features that can influence the speed at which this electrical signal is conducted down an axon. So the speed of an action potential increases with the axon's diameter. So sodium ions that have entered the axon exterior at the start of an action potential are going to, remember, repel those intracellular cations, those other positively charged particles that are already inside, and cause them to spread along the inside of the membrane. That's what's going to allow the signal to propagate. So these cations are better able to move. They meet less resistance in axons with large diameters than those in narrow axons. So a larger diameter axon increases the speed of propagation of the action potential down the axon. So the charge spreads along the membrane more quickly with less resistance. And the best example that we have of an axon with very large diameter is the giant axon in the squid. And so a lot of the research that's been done that allows us to know what we know about action potentials were done on these squid giant axons that you see going through the mantle. And they're very long, and they also have a very large diameter, so they can be easily manipulated and measured. In vertebrates, we do something a little bit different. We tend to have very small diameter axons, but we have them insulated by something called a myelin sheath, which causes an action potential speed to increase. So myelin sheaths are made by glial cells. In the central nervous system, these glial cells are called oligodendrocytes. In the peripheral nervous system, they're known as Schwann cells. And these glial cells sort of wrap in several layers. They kind of spiral around portions of the axon. And these spaces in between these kind of wraps of glial cells are known as the nodes of Ranvier. And it's only at these nodes of Ranvier that are not covered with myelin that you find these voltage-gated sodium channels. So they're restricted to the nodes of Ranvier, which are the gaps in the myelin sheath. So think of an individual cell kind of wrapping and spiraling around an axon, another one next to it is going to wrap and spiral around, and then there's going to be a little gap in between those two. And that's where the voltage-gated sodium channels are located. So action potentials in myelinated axons are going to actually jump between the nodes of Ranvier in a process called saltatory conduction, almost as if those portions of the axon that are coated in myelin did not even exist. So let's see how this works. Where this axon is myelinated, where this Schwann cell is wrapped and wrapped and wrapped around it in many layers, this is going to prevent ions from leaking out. So there are going to be none of these leak channels where ions can escape in this region. So when there's an action potential here at this portion of the membrane, you're going to have this influx of sodium ions, which are going to repel all of the cations in this area. And so all the cations here are going to be moving in this direction, which is going to cause cations a little bit further down to move. And this is going to happen almost instantaneously because everything is sort of pushing outward simultaneously as cations are repelled from this area. And so the action potentials are going to be regenerated at the next node almost instantaneously as those cations are pushed away. It's going to depolarize this portion of the membrane and cause an action potential, which again is going to cause this influx of sodium ions, which is going to spread to the next one. At this point, this portion of the membrane is repolarizing. The sodium channels are blocked. They're going to be insensitive to the flow of cations from this direction, but this one is going to be ready to be activated, and it's going to jump from one node to the next very, very, very quickly. So signals continue to jump down the axon in this way, propagating more quickly than it would down an unmyelinated axon. So now let's think about what happens when this action potential reaches the very end of the axon. So there are a couple of different kinds of synapses. One is called an electrical synapse. And in this case, the electrical current flows from one neuron to another through gap junctions. So you don't have this chemical signal. The action potential just continues to propagate from one neuron to the next. And this happens in situations where a very rapid response is needed and a very coordinated response, and it's relatively rare. Most synapses are chemical synapses in which a chemical neurotransmitter carries that information between neurons through this small space in between called the synapse. And so most synapses are going to be of this type, a chemical synapse. So these are the ones we're going to examine in detail. So what happens when an action potential arrives at the end of an axon? So most neurons are separated from one another by tiny spaces called synapses. So how do we know how this message is transmitted across the synapse? How do we know that it's a chemical messenger? We've known this actually for some time. In the 1920s, a scientist named Otto Loewy did some experiments with frog hearts that showed that these messages are actually transmitted chemically. So here is the experiment that he did. He took a frog heart and artificially stimulated the vagus nerve that goes to the heart. So the vagus nerve is part of the parasympathetic nervous system. When it's stimulated, it's going to tend to have a calming effect. Remember, this is the rest and digest part of the nervous system. And so when the vagus nerve is stimulated, the effect is to slow down the heart rate. They stimulated it, saw that the heart slowed down. After that stimulation, they collected up some of the fluid surrounding that nerve ending and took a second frog heart and just exposed it to the fluid that was taken from the stimulated heart. So this is not being stimulated in any way. And so if there's a chemical signal that's causing this response, you would expect the second heart, even in the absence of an electrical signal, to also have this response. And this is exactly what he found. In the second frog heart that was just bathed in the chemical solution but had no electrical stimulation, there was also a slowing of the heart. This shows that there's some chemical in the fluid that's actually producing that response. So this supports the hypothesis that the signal from cell to cell is delivered by chemicals, and we call these chemicals neurotransmitters. So these are the chemical messengers that transmit information from one neuron to another, or to a muscle or a gland. So the membranes of axons and the cells they communicate with are separated by a tiny space known as the synaptic cleft, this little gap right here in between. And the ends of axons contain these little vesicles, these little membrane-bound vesicles called synaptic vesicles that store the neurotransmitters. So the neurotransmitters are synthesized within the presynaptic cells. After they're synthesized, they're packaged in these vesicles, and they are sitting in the axon terminal waiting to be deployed. So let's take a look at how this happens. So this happens in several steps. So the first thing that happens is the action potential is propagated all the way down to the very, very end, down here at this synaptic junction. And when it arrives at the end of the axon, the depolarization here at the end is going to cause the opening of voltage-gated calcium channels. So instead of the sodium channels that were propagating this action potential, now we're going to open voltage-gated calcium channels as a result of this depolarization. And these are going to be located right in the region of the synapse, and the calcium is going to flow into the cell along its electrochemical gradient. So in response to that influx of calcium, the synaptic vesicles are going to move to the cell membrane. So remember, these cytoskeletal trackways, the calcium is going to activate these motor proteins that are going to bring these vesicles to the cell membrane. They're going to fuse, and when they fuse with the cell membrane, they're going to release their contents, the neurotransmitters, into that synaptic cleft by exocytosis. So now, as we've seen before, when we talked about graded potentials, those neurotransmitters are going to bind to receptors on the postsynaptic membrane, acting as a ligand. And remember that ligands refer to molecules that bind to a specific site on a receptor molecule. The binding of the neurotransmitter leads to a change in the membrane potential. Remember, they're going to open these ligand-gated ion channels, and then the combined effect of many neurotransmitters may or may not trigger an action potential in the postsynaptic cell. Finally, we need to shut this response off at some point, and usually these signals are very, very brief. So the response ends as the neurotransmitters unbind from the receptors. So remember, these ligands are always binding reversibly, binding and unbinding, and they're going to do this repeatedly until they're removed. So the way to stop them from binding and stop this action that they're creating is that they need to be removed from the synaptic cleft. So either they're going to diffuse away, they can be broken down by enzymes, or they can be taken back up by the presynaptic cell to be used again, repackaged in vesicles, and reused. So let's take a closer look at how that happens. So here is the release of the neurotransmitter. This vesicle moves to the cell membrane, fuses with it, opens up and releases the neurotransmitter. And on the postsynaptic neuron, in the case of enzymatic breakdown, there are both receptor proteins and also these degrading enzymes right in the membrane that are going to break them down and make sure that this signal does not last for very long. Why we want to make sure that it takes a continuing signal coming from the neuron to produce a continuing response in the postsynaptic membrane. We don't want this stuff hanging around. In this case, the neurotransmitter is being released, And the presynaptic membrane has active transporters for the neurotransmitters. So as it's being released, some of these are going to come and bind with their receptors. And when they come close to these active transporters, they're going to be brought back in, repackaged into vesicles, and ready to use again. So we've been thinking about these neurotransmitters as binding just with these kind of ligand-gated ion channels that are going to directly open up the ion channel. But in some cases, there are more steps involved, and we call these neurotransmitter receptors metabotropic. And so let's take a look at the different steps that this involves. So in this case, movement of ions through a channel depends on one or more metabolic steps. So it's not going to happen immediately as a result of the ligand binding. There are going to be more steps. Binding of a neurotransmitter to a metabotropic receptor activates a signal transduction pathway in the postsynaptic cell involving a second messenger. So very similar to all of the signal transduction pathways we've been looking at so far. So the signal transduction pathway leads to an amplification such that many channels can be opened or closed in response to a single binding of a single neurotransmitter. So many neurotransmitters have both ionotropic, so the direct ligand-gated channels that are going to open just in response to the binding of the ligand, and also metabotropic receptors that are going to go through this amplification second messenger pathway that's going to allow a much larger response to a much smaller quantity of neurotransmitter. So let's briefly meet the different kinds of neurotransmitters that we commonly see in vertebrate nervous systems. So a single neurotransmitter may bind specifically to more than a dozen different receptors. The response that postsynaptic cell is going to have to that neurotransmitter is going to depend on what kind of receptor it has, because remember, the receptor is the ligand-gated ion channel, and that's going to be specific to one particular ion. So a single neurotransmitter could excite postsynaptic cells expressing one receptor and inhibit postsynaptic cells expressing a different receptor, depending on whether that receptor is a sodium channel or a potassium channel or a chloride channel. It's going to be excitatory if it's a sodium channel. It's going to be inhibitory if it's a potassium or chloride channel. So acetylcholine is a very common neurotransmitter in vertebrates and invertebrates. I don't want you to necessarily memorize all these different neurotransmitters, but what I do want you to remember is that acetylcholine is the neurotransmitter of the neuromuscular junction in vertebrates. So acetylcholine is involved in muscle stimulation, but also in memory formation and also learning. So vertebrates have two major classes of acetylcholine receptor, one that's ligand-gated and one that's metabotropic. So the ones at the neuromuscular junction are ligand-gated. The ones that are involved in learning are metabotropic. So a number of toxins disrupt acetylcholine neurotransmission. So for example, the nerve gas serin inhibits the enzyme that breaks down acetylcholine so that acetylcholine remains in the neuromuscular junction and causes constant, uncontrolled contraction of muscles. Botulism toxin paralyzes muscles and is produced by certain bacteria. And acetylcholine is just one of more than 100 known neurotransmitters, and these fall into a couple of major categories: amino acids, biogenic amines, neuropeptides, and gases. So glutamate is one of several amino acids that can act as a neurotransmitter in both vertebrates and invertebrates. Glycine acts at inhibitory sites in parts of the central nervous system outside the brain, so in the spinal cord. And gamma-aminobutyric acid, more commonly known as GABA, is the neurotransmitter at most inhibitory synapses in the brain. So what I do want you to remember is that glycine and GABA are the major inhibitory neurotransmitters. They're going to tend to hyperpolarize the membrane. Biogenic amines include norepinephrine, epinephrine. Both of these two are involved in stress response, dopamine, and serotonin. So biogenic amines have a central role in a number of nervous system disorders. For example, Parkinson's disease is associated with low amounts of dopamine in the brain. There are several neuropeptides, so these are short amino acid chains that also function as neurotransmitters. Neuropeptides include something called substance P and also endorphins, which both affect our perception of pain. So you've probably heard of endorphins. Opiates bind to the same receptors as endorphins and can be used as painkillers. They also produce a sense of euphoria. Gases such as nitric oxide are local regulators in the peripheral nervous system. Unlike the dissolved neurotransmitters, it's not going to be stored in these membrane-bound vesicles. It's actually going to be synthesized as it's needed and also broken down very quickly after its production. So it's going to be a very transitory kind of signal. And not just nitric oxide, but also carbon monoxide is used in very small amounts as a neurotransmitter. So we all know the dangers of inhaling carbon monoxide, but your body synthesizes it, and some of that is used as a neurotransmitter to transmit these neural signals. So far we've been talking about just how these messages are transmitted from one neuron to another or from a neuron to its target. And I gave you this framework for the organization of the vertebrate nervous system. So as we move forward into the next section, we're going to be focusing on the sensory part of the nervous system, how we obtain information from our environment. So both special sensory, smell, vision, hearing, and taste, the senses associated with the head, and the general sensory, the senses that are distributed throughout the body, touch and stretch, reception, and how these signals are received by sensory structures, how that signal is transduced and converted into this electrical signal that we've seen transmitted throughout the body. So all of these senses that we use all the time to interpret our world are converted into these electrical signals. So in the next couple of web lectures, we're going to see how that happens.